Welcome to this biochemistry lecture on RNA. So most of us have a pretty good handle on DNA. I'd say less numbers of us have a good handle on RNA. DNA is used to store genetic information. RNA is as well. Double-stranded DNA is the norm. Single-stranded RNA is the norm. And basically we're going to, as noted in the central dogma discussions, double-stranded DNA is transcribed into mRNA. mRNA is then processed and then is translated into protein. And it's one more mechanism by which we can regulate what proteins are produced. So double-stranded RNA actually, if we have a double-stranded RNA, is not translated. And so there's some regulation elements that are going into RNA that are really important. Again, double-stranded DNA, our DNA is usually double-stranded. It's usually an alpha helix in a B form, and it's usually supercoiled. RNA is usually a lot shorter. It has numerous different functions. Double-stranded DNA usually signals infection, it mediates editing, and it's used to silence genes. There are many structures of RNA and that include different functions. They're important in enzymes, including the ribosome. They are important at binding sites in the scaffold. They're signals, and also they're templates for telomeres. So those are the important differences between RNA and DNA. As far as the structure goes, there's only one atom that's different between RNA and DNA. And if you look on the right-hand side over here, you can see that in a ribose, there's an alcohol subgroup, whereas in a deoxyribose, there is just a hydrogen. And so that hydrogen, that change from a hydroxyl to a hydrogen on that 2' group allows for significant difference in structure and thus function. And everything else is very similar. This slide gives a really good description of where the nucleoside, where we have the nitrogen base and the ribose, whereas a nucleotide we add in the phosphorate linkages for onto a nucleoside. Those are very comparable structures between RNA and DNA. There are three major types of RNA. There are numerous subclasses. The three major types are mRNA, ribosomal RNA, and transfer RNA. And we'll talk about all three of these. Messenger RNA is used to translate, or is the template when we translate RNA or into protein. Ribosomal RNA is part of the structure of the ribosome, and that is where the translation occurs. And tRNA take amino acids and add them to the growing polypeptide chain. So those are the major forms of RNA, although there are numerous other types of RNA that exist. So here are the other species, and this is just a good reference tool. We'll talk about the top three. We're not going to talk about small nuclear, microRNA, and small interfering. Those are all used as processing and for gene expression, and they're important, but they're not beyond the scope of what we're going to talk about today. So messenger RNA comes out of the nucleus, and codons come in, and they have the tRNA, which we'll talk about here in a minute, bind to the messenger RNA in triplicate form. The appropriate one does, and that's how the growing polypeptide chain grows. And so it's important to note that that's the function of messenger RNA. So there's several RNA structural motifs, mRNA structural motifs. Those are important in regulations. Most of the time they're just single strand. There are double strand that is then not translated. You can have single nucleotide bulges. You can have hairpin loops. You can have symmetric internal loops. You can have asymmetrical internal loops. You can have 3 nucleotide bulges. All of these exist in secondary and tertiary structure, and all of them have important regulatory and function. So the tRNA, which you'll see here in a minute, has a specific function, and so does rRNA based on these structural motifs. And they do hybridize very similarly to DNA with hydrogen bonds in between the between the nitrogen bases of the nucleotides. So these structural motifs do provide important structure and thus function. As RNA is processed, this is going to be an important part of the discussion. There are several, so DNA goes to pre-RNA before it's processed. Once it gets to the transcription level, we're going to add a 5' cap right here, which we've discussed in other lectures, and a poly A tail. Also, after that, then all of the introns are removed and exons are left remaining, and then it gets translated into DNA, or into protein. sRNA is a small nuclear RNA. It's important in this processing. The 5' cap is actually a methylated GTP, and it makes translations significantly more efficient. The poly A tail is a modification on the 3' end. We add several hundred A's. It protects this from the mRNA from degradation, and the splicing again is important to form very unique. So we can take a gene, we can splice it differently and get different proteins that are for the same family out of that one transcription. These are highly regulated. snRNAs, miRNAs, and siRNAs all are important in regulation and in part of RNA processing. Again, a single gene can encode several different types of proteins, and then the exons that different exons will dictate. And a classic example, this is antibodies, where we can get isotype switching, or you can get going from a plasma cell to from plasma where it's expressed on the surface of a B cell to a mature B cell where the antibody is just being spit out. This is a classic example of where exon splicing dictates the structure of a cell. The antibody is the same. It's just how whether it's anchored in a cell or whether it's globular and floating around in the cell. That's all done by exon splicing, and so it's an important part of RNA processing. tRNA, again, this is the structure, and this three-dimensional structure doesn't really do it justice. You have an RNA and important elements of the RNA are the anticodon loop, and you also have the CCA terminus where the amino acid then attaches, and so this tRNA brings in the amino acid into the ribosome, and it hybridizes to the mRNA at the anticodon loop at the end. Then once it hybridizes, then you're going to add in the volume for the growing peptide, the polypeptide. Again, our next is the rRNA, which is the ribosomal RNA. Interestingly, in our ribosome is about 2/3 RNA and 1/3 protein, and there's two different subunits, and they both have very important function. And the function is the enzymatic function of adding new amino acids to this growing polypeptide chain. That's what the rRNA does, and the ribosome is composed significantly of these rRNA subunits. That's the important discussion of RNA. I hope you enjoyed, and we look forward to seeing you in the next round.